Dr. David Goldstein, who is going to talk to us about the TRPV4 mutants. He comes to us as the founder and CEO of Actio Biosciences.
Many thanks to Ryan and the other organizers for the invitation to be here, and hopefully, maybe I have to do something for the slides. Hopefully the slides are coming up. Maybe just go back one. Or we can start on that also. So it's a pleasure to be here. I'm going to be introducing our inhibitor of TRPV4 0871. And I'm really excited to share that we're announcing here, in fact, for the first time, that this program will be entering clinical testing later this year. So I'm going to get to an introduction to the program very shortly. But before I do that, just a quick introduction to the company and how we try to work. So we're a biotech in San Diego, founded just under 3 years ago. The basic strategy for the company is to develop truly transformational medicines for rare diseases with very high unmet need. But by working in a way that allows us to truly understand the biology of what we're trying to do. And this goes back to a point that Rob made. Even if you're targeting mutations that cause disease, you often don't know exactly what to do unless you really understand what the mutations do to cause disease. And that actually is very challenging, much more challenging, I think, than a lot of people realize. So I'll really try to emphasize that point a little bit later in the talk. The other aspect of what we do beyond targeting rare diseases with very high unmet need is try to learn from these rare diseases things about the basic biology of the target that gives us pointers to where else the target might be relevant beyond the disease caused by mutations in the target. I won't be talking about that today, but that's a fundamental part of our strategy. Just a little bit of detail about how we actually work. What we do is first identify a target where we believe we can truly understand the underlying biology. And then we really develop the preclinical tools to make sure that we do understand the underlying biology. That means that we characterize the functional effects of the mutations, believe that we have to believe we're looking at functional effects that relate to how the mutations actually cause disease. We have to be able to characterize construct valid and face valid preclinical models, meaning that the underlying cause of disease in the model is similar to what happens in humans, that the presentation looks like what happens in humans. That allows you to test therapeutic hypotheses, such as whether you can have an impact on symptoms after they emerge. And then we begin, as I said, exploring connections between the target and other indications. So now, to move on specifically to the TRPV4 0871 program. So it has been mentioned earlier, and as is known by most here, mutations in TRPV4 cause a rare form of CMT called CMT2C. The mutations that cause disease are activating in nature. We have reassured ourselves about that, and I'll say more about that in a bit. There are about 60 pathogenic mutations that have been reported and characterized. And the presentation of CMT that results from these disease-causing mutations in the target result in a particularly severe form of CMT with some of the more typical features of CMT, but also some distinctive features, such as a pronounced focal cord involvement and some other features, including, for example, in some cases, a particularly severe form of overactive bladder, which does actually provide a pointer for where the target, other places where this target may be relevant. And I'll be describing our first-in-class inhibitor, ABS 0871. So first, to try to understand the preclinical model. We have been working with Charlotte Sumner at Hopkins, Brett McRae at Hopkins, Jeremy Sullivan, and others to understand the details of what the preclinical model looks like, what treatment in the model looks like, and something about the underlying biology of how mutations in the target actually cause disease. So the way this work is done is you build a model around one or more of the mutations that cause disease. In this case, it's R269C. This model has a pronounced phenotype with real muscle weakness, and if we play the video, you can see what it looks like. We can go ahead and try to play that. This is what the mouse looks like pre-treatment, so you can see that it's not mobile. The mouse should be running around the cage. It's not, so you can see evidence of pronounced weakness. After treatment with our inhibitor, we get what looks like complete reversal of the symptomatology. I should note that if you treat later in disease course, the reversal is not as pronounced as that, though there is still improvement. That, of course, is surprising that you're not here looking in the preclinical model at arresting the progression of the symptomatology, but you're actually looking at reversal of the symptomatology. The team at Hopkins has done a lot of work to understand why you get this reversal, and it turns out that this is a very distinctive mechanism for how you get CMT, where what's actually happening is that the activated channel is causing disruption in the barrier between the vascular and the nervous system. There's direct experimental evidence of that. So when you have the activated channel expressed in vascular endothelial cells, you get leakage out of the vascular system, compromising neuronal behavior. After treatment, the leakage actually stops, and the neurons apparently very rapidly recover their normal behavior, and that reverses the symptomatology. So this is a really strong, really good indication that we might be able to not only arrest progression of symptomatology in this disease, but we might be able to actually outright improve the symptoms. The other thing that we always try to think about in the company is being able to treat all the patients that have disease because of mutations in the target. So in order to do that, what we do is functionally characterize literally every reported disease-causing mutation. As I said, there are about 60 of them. This is really hard work to do because you have to find an in vitro model where you can actually get the protein to do what it's supposed to do. So this is a calcium channel, as the earlier slide indicated, that's expressed in the cell membrane. In some cell systems, it's difficult to get the mutant form of the channel to the cell membrane where it's supposed to do its work, so you have to sometimes explore different cell types in order to do this work properly. So we've been working at this for a good long while. For the majority of the mutations, we've been able to get them expressed and be able to run an assay in appropriate cell type to show the activating effects of the mutation. I do want to make one point here. It goes back to a point that Rob made. It really is the case that disease-causing mutations do lots of things. It's just the rule. And so what that means is if you take a few disease-causing mutations and you put them in one functional assay or another, you'll often see functional effects in these very artificial in vitro assays that have absolutely nothing to do with the way the mutations cause disease. So it's really challenging to do enough work to convince yourself that you understand the way the mutations cause disease. One of the ways that you do that, not the only way, but one of the ways you do that is you characterize lots of mutations and look for consistent effects across the full range of mutations. Then you start to gain confidence. In this particular case, we really do now know what's happening because we have characterized enough mutations to show consistent, for all of them, but for most of them, consistent activating effects. And of course, we've got the preclinical model where inhibition normalizes. So we really do believe that we are looking in these assays at effects of the mutations that relate to how they cause disease, meaning that what we want therapeutically is indeed an inhibitor. But in any event, as you can see in the pie chart, for the majority of the mutations, we've been able to show activating effects, and for a minority of them, we've not been able to run an appropriate assay. Now, I do want to say that that does not, in our interpretation, mean that for those other pathogenic mutations that they're not activating in effect. It means we can't assay them in the various cell types that we've tried. So for the ones that we can assay, the majority of them, What we then do is ask whether the inhibitor that we've developed actually shows activity against all of those mutations. Because we can actually show that we inhibit the wild type, but that's actually not going to be how you treat disease. So what we've done is assess potency of our inhibitor against all of these mutations that do have activating effects, and you can see the result on the right-hand side. What you're looking at are IC50s for the wild type on the left in the figure, and for all the assayed mutations on the right. And you see that there's a very modest spread of activity across these different mutations. And so what that means is that we will be able to get to sufficient exposures in patients to inhibit these different activating mutations, and indeed actually normalize the activity of the aberrant channel. And this is an absolutely critical point, because what we'll actually be doing when we're treating these patients is bringing the activity of the channel back to normal levels. It's completely targeted to the underlying cause of disease, again, a point that Rob made earlier. And a way to look at that is that actually has to have a therapeutic benefit unless the disease is completely developmental. And in this case, there's no indication that it's completely developmental, as the preclinical results show. Partially developmental, of course, not completely. So we are now progressing, of course, ABS, as I mentioned, ABS-0871 towards the clinic. It will be in healthy volunteers. The plan is that it will be in a healthy volunteer study later this year. And we are thinking now, of course, about appropriate endpoints for the pivotal trial when that trial is run. One really exciting possibility for an endpoint is focused on the vocal cord dysfunction that the majority of these patients do show. And one attractive aspect of the vocal cord dysfunction is that it can be accurately quantified with laryngoscopy, and this can actually be accurately quantified in all patients regardless of the severity state, so that will really help us get a consistent measure across all patients. This and other aspects of the presentation is now being carefully characterized in a natural history study that Brett, who is here, is involved in, along with Charlotte and others at Hopkins, and Brett's now at University of Michigan, and we'll be talking later today. And we, of course, are also looking at other potential endpoints, including ones that are more standard instruments in CMT more generally. The overall development plan is on the right-hand side with the healthy volunteer study starting later this year, and then the plan to move next year into a small 1B study in patients, give us an idea of what aspects of the disease presentation might change over what time course, and then the learnings from that 1B would be taken forward into a phase 2 study that would in fact be the pivotal registrational study. So that's the program, that's where we are. I really want to emphasize that you don't develop a program for a rare disease without a whole community of people working on it. We've been really privileged to work with some of the really strongest people in the field working on TRPV4 and CMT2C, some of whom are here. And the company, as I mentioned, is based in San Diego. We are still recruiting. It's a wonderful place to work, so please let me know if you have any interest, and thanks very much for your attention.
Well, I think we have time for maybe one or two questions.
Very nice talk. I have maybe two questions. One, could you comment on off-targets to other calcium channels and how you counter-screen to the extent you can? And two, is the excitability of the axons affected, and could you in humans measure that as a
So the first part, so the first question, we have done lots of selectivity assays. The compound actually doesn't have any meaningful activity against any of the other TRPs and lots of other things that we've assessed. The TRPs, as you probably know, are not all that closely related, so not terribly surprising, but no activity there otherwise. Then the next question I didn't quite hear.
Curious if the resting excitability of the axons is altered, and could you measure it in humans?
Yeah. So we actually haven't looked to see how so the first thing to say is that this is not a cell autonomous effect within neurons for sure. So if you, in the preclinical model, if you actually take the activated channel out of neurons, it's modestly expressed in neurons, not that highly expressed, but if you take it out, you get the full disease presentation. So it is not a cell-autonomous effect in neurons. So we don't think there's any relevance really to what's happening in neurons. If it's knocked out of the vascular endothelial cells, you get complete rescue. So it really is driven by what's happening from leak. There are some electrophysiological signals that you can see in patients, so the neuronal behavior is affected, but it's a secondary effect.
Other questions? Will's got one here.
Thanks. So if your natural history study confirms that you really never see improvement, and in your 1B you do see improvement, do you think there's a registrational path to approval without doing a placebo-controlled study?
So if our 1B, so I didn't hear the first part.
If the natural history study shows that you don't see improvement in the natural history of the disease, but you do see
improvement, what do you mean in the natural history
study? Like you only
'Cause-
see decline, but then when you
But there's
treat
no
patients-
intervention, there's no intervention
exactly.
in the natural history study.
Right, but then when you treat in your 1B, you do see improvement
Yeah.
in clinical and physiological measures. Do you think that there's a registrational path without doing a placebo-controlled study?
Oh, I see, without placebo control. We're actually not really right now planning on not using placebo. We are planning to use a placebo control. Even if we took the view, so I think I understand the question now, if we took the view that, which I think it's better to be cautious and not, but if we took the view that we might expect to see equally dramatic reversal when we run the trial as we see preclinically in some of the patients, you might see your way to saying that you could have sufficient efficacy for approval without the placebo arm. However, there still is a lot of advantages to having the placebo arm to actually assess safety. So right now the plan is in fact to have a placebo arm in the pivotal.
Okay, well, I think we should probably go ahead and move on. Thank you.